Hello, everyone. Good afternoon. My name is Allie Hines, and I'm the Senior Manager for Communications at Providence Healthcare in Eastern Washington. And we have a very important topic that we're talking about today with some of our top experts on the matter. Of course, we are talking about COVID variants and mutations, what we know so far. And joining me today is Dr. O, the laboratory medical director at Providence Everett, and then also Dr. Chong, our Chief Medical Officer for Providence St. Joseph Health System Laboratory Services. So thank you both for joining us today. Before we begin, we do have to go over a disclaimer that the information provided during this event is for informational purposes only. This event does not create a doctor-patient relationship, and any questions or medical advice discussed is not considered guidance on what you should do. For medical questions, please reach out to your primary care or healthcare professional. And so now that we have that out of the way, let's go ahead and start, starting with our introductions to getting to know our panelists, our experts. Dr. Chong, can you tell me a little bit about yourself?
Hi, good afternoon. I'm the Providence Chief Medical Officer for Laboratory Services. Providence has more than 50 hospitals in 7 states, and I coordinate the hospital labs. I also advise the 1,000 clinics that are in Providence on laboratory testing.
Thank you, Dr. Chong, and turning it over to you now, Dr. O.
Hi, I'm the Medical Director at Providence Regional Medical Center in Everett. I've been in this position for about 3 years, and I also help coordinate the regional lab services for the Washington, Montana region.
Thank you so much both for joining us today. And we're talking about a topic that a lot of people have questions about. They're hearing on the news that there's new COVID variants and mutations. So let's just start off by, what is a mutation? What is a variant? And how do they even occur?
Okay, so mutations are a natural byproduct when the viruses multiply. The SARS-CoV-2 is an RNA virus, which means its genome is encoded in RNA. And RNA viruses such as SARS-CoV-2, HIV, influenza viruses, they tend to pick up mutations quickly because the enzymes or the polymerases that copy its RNA genome, they're prone to making mistakes. But data from the University of Bern in Switzerland suggests that actually the SARS-CoV-2 virus, which is a coronavirus, they make fewer mistakes than most other RNA viruses. And that's because they have a proofreading enzyme that corrects potentially fatal copying mistakes. So they correct the mistakes before it hurts the virus. And a typical SARS-CoV-2 virus, it accumulates only about 2 single-letter mutations per month in its genome, which is a very low rate. I mean, it's a rate of change that's about half that of the influenza virus and 1/4 that of the HIV virus. And yet, despite its sluggish mutation rate, researchers have cataloged more than 12,000 mutations in the SARS-CoV-2 genome so far, and that keeps adding up each day.
Thank you, Dr. Chong. And Dr. O, if I remember correctly, you have been studying the current COVID mutations and variants that are out there. Can you tell us about what we know about them so far?
So these variants, some have no consequence to the actual virus's ability to transmit or produce severe disease. However, some do produce mutations that have advantages for the virus, both in transmissibility, or they're more virulent or cannot or are able to escape the current treatments we have for it. And this is really why this subject has come to international importance right now, is because as we come up with more treatments for the virus, and the vaccines are very specific in some instances for very specific areas on the virus, these mutations have the potential to affect how our current treatments can really fight against it. So right now we have variants of concern, and really the terminology is all over the place. There's lots of letters and numbers, and it's very confusing. But Specifically, the variants of concerns are mutations that change and have survival antigens that will go on and multiply and spread. Now, some of these mutations occur in the same branch point of the tree. However, some of them arise spontaneously in completely different branches of the phylogenetic tree of the SARS-CoV-2 virus, and each of these mutations can cause a change in the virus proteins. Some of them are in key areas of the spike protein, which is the binding site for the virus to human cells, and they've been shown to decrease the effectiveness of monoclonal antibodies or convalescent plasma that has been derived from mutations to previous variants. A lot of these are being discussed in the news, like B117 or B1351, and have several mutations. And some of them are overlapping and some of them are not. That's really why they're coming to the forefront of research at the current time.
And Providence is tracking these, correct?
Yes, we are. Well, I mean, we do track the literature, but then Providence also has a genomics lab in Portland, Oregon. And Providence has 6 hub labs in the 7 states that we have hospitals in. So we have hub labs in Anchorage, Spokane, Portland, Burbank, Orange County, and Lubbock. And so they do submit samples to our genomics lab for sequencing. And then we have 45 other hospital labs that do more acute testing, but they are also on the alert for any outbreaks and unusual pattern. And they will also submit positive samples to Portland for sequencing. And we coordinate the laboratories in Providence on a weekly virtual command call. And I have a systems director who compiles a weekly report of the number of tests we do, how many are positive. And so we have an idea whether we have unusual patterns. And of course, finally, Providence has a very good analytics team led by Dr. Ari Robichak that tracks hospital census by geography, by ministry, and that information is shared weekly with the entire Providence team.
You mentioned sequencing. I'd like to talk a little bit more about that. What is it, and why is that useful?
So viral sequencing is periodic sampling of viruses in the community and really looking at the entire genome to track the prevalence of certain strains and monitor the development of mutations. Previous to this, throughout the pandemic, there have been multiple academic articles looking at tracking the family tree and the branch points, and it was used not only to identify new mutations, but also track the progress and spread of the virus through different countries and how they evolved in different environments. It's key to being able to provide information to help direct controlling measures for the epidemic. For example, the AstraZeneca vaccine was pulled from South Africa because of concerns the vaccine would not provide enough immunity against the most prevalent variant there, which is the B1351 variant.
Is there a difference between sequencing and surveillance?
So sequencing is the actual testing, and surveillance is compiling that data and really looking at the population trends of where you're seeing those mutations arise. There's been a large amount of ebb and flow in different mutations prevalences throughout the globe. And tracking that is important to being able to understand the patterns of disease and how different countries can respond to it.
I'd like to go back to Dr. Chong because he was talking about the research. What research is being done in Providence labs toward understanding mutations?
Well, so the genomics lab, have been sequencing samples, the positive samples we get from the different hub labs, and so we actually confirmed that the California Cal.220C that was reported actually just published on February 11 in JAMA that yes, we actually found that particular variant in our samples. In fact, in 30% of the samples submitted from LA County, Providence hospitals were of that variant, and 65% from Orange County were also of that variant. So, and that was the variant that was caused a surge in Southern California in November, December. So we did actually find those. And the research effort in Providence is actually a close collaboration of our ID doc physicians, our genomics lab, our clinical labs, and our analytics team. The leaders are Doctors Walter Erber, Jason Goldman, Josh Diaz, Brian Peening. And the Dr. O and I coordinate the clinical laboratories. And we can contribute a lot to the knowledge pool out there because, in addition to identifying just the mutations, we have the clinical protocols, we have the history, we know the outcomes, we know how much oxygen the patients need, uh, whether the treatment work, when they're given monoclonal antibodies or convalescent plasma. We know their vaccination status. So besides just knowing the mutations, we actually have epidemiology and clinical data. And that makes the research, our data so much more richer and powerful. And it's all in our electronic medical records. So Providence, we can contribute a lot to the knowledge of these mutations and variants.
I imagine that having this collaborative effort is very beneficial for us, and especially having them in different regions. Have you been able to see how, and if so, how do mutations differ from region to region?
Well, we are actually now we are taking on a bigger step in us collecting more samples because for the hub labs were actually set up not too long ago. I think some of them came up in August, but most of them actually were more operational in November. So our effort has been fairly recent. But now that we have the hub labs really fully operational, we'll start this next phase of collecting and sending more samples to the Oregon lab for sequencing. And then if we find any unusual pattern or new ones, we can look back in the electronic medical records. The ID physicians will, of course, be doing that and analyzing the data and trying to explain, okay, which ones did impact that clinic, which one produced maybe worse disease. So we will be analyzing all those aspects of the clinical cause.
In your research, have we seen if mutations affect different age groups differently?
So the most well-characterized international variant is the B117 variant, and there's been recent data that's been coming out of the UK showing that, in fact, the younger age population has a higher prevalence of disease and is being infected at a higher rate by the B117 variant than previous strains. However, it is fortunate to note that the survival curves are not different in the younger age group in those patients who are infected with the new variant. That same cannot be said for the older age group. There have been several reports out of the UK looking at this variant, and there's increased severity of disease in the older, above 70 age population. So there has been alerts going out from the UK government with this information.
I think that the two big news items right now in relation to COVID is about variants and about vaccines. So let's switch over now to vaccines. Do we know if vaccines, the current vaccines that are out, will help protect against the new variants? And if so, why or why not?
So currently, vaccination studies and overall studies looking at the efficacy of these vaccines are really centered around neutralization studies. And that's where researchers simulate a virus by introducing specific mutations in their RNA sequence, popping that into a pseudovirus and using that to infect a strain of cells. And that strain of cells are looking at whether or not that altered virus can infect the cell, and if they add antibodies to it, will those antibodies prevent infection of those test cells? right? So those are neutralization studies, purely looking at the antibody neutralization ability of these different antibodies. And it can be convalescent plasma raised against previous strains of COVID-19. It could be monoclonal antibodies, or it could be antibodies derived from patients who are vaccinated from different vaccines. And what the data is showing now in the literature is that against the B117 variant, that the vaccines from Pfizer and Moderna, for example, show slightly decreased but fairly good neutralization activity against these cells. The neutralization activity against the B117 with another gene showing high transmissibility and possibly severity called E484K shows slightly even more decreased neutralization activity. And in comparison, the South African variant shows even greater decrease in neutralization ability from the convalescent plasma of previous strains. And so that in itself is a little concerning, though we know that any immunity is better than no immunity. If we turn to looking at monoclonal antibodies, there are several different papers looking at the ability of monoclonal antibodies to neutralize these different variants. And what's been shown is some of the monoclonal antibodies that are in production or research completely cannot neutralize the South African or the B1351 variant at all. Okay, then we move on to the vaccine sera, and these are very early studies. They're pretty early and they're not huge numbers. But again, for the Moderna and the Pfizer vaccine, their efficacy against the B117 variant is decreased, but still pretty good. And slightly more decreased in terms of ability to neutralize the B1351 variant. The very interesting thing is the AstraZeneca vaccine. So that vaccine is slightly different than the first two vaccines, and it was tested out in South Africa. And the South African government actually pulled that vaccine out because in their efficacy studies, looking at the frequency of infection in a placebo group or a group of patients who had received two doses of the AstraZeneca vaccine showed that they're basically overlapping. There is a maybe 20% efficacy of that vaccine against the B1351 variant, which is the predominant variant in that country. And so they are switching to a different platform. It's sobering, but there's already a lot of discussion in the literature about using monoclonal antibody cocktails, about getting booster shots. AstraZeneca is looking at changing their formulation of their vaccine a little bit, or of moving to something like what we saw in the influenza vaccines, which is divalent, trivalent, quadrivalent vaccines, looking at including many different types of SARS-CoV-2 viruses in that vaccine formulation in order to increase its efficacy.
There's also the question of people who have already had COVID. If they've already had COVID, could they be infected by one of these variants or mutations?
Yes. So in the past, the estimates for previous infection efficacy protecting you against the, strains that have gone before has estimated but anywhere from like 80 to 95%. That is, like, if you had COVID-19 before in March of 2020, you would have maybe a 5% chance of getting it later on in October of 2020, something like that. It's not entirely clear what that reinfection rate will be with the increased mutation rate. I think it's concerning to everybody that rate could go up. The original thought was that if you've had it before and you had some sort of immunity to it, then if you get it again, it might not be as severe. And that is certainly the hope, I think, for everybody. It may not hold true in patients who are immunocompromised or older, and I think it really remains to be seen what that efficacy. the studies I was talking about are all neutralization studies, only looking at antibodies. Okay. And the rest of our immune system is still going to be involved in producing a response against these viruses. So those are studies that are still ongoing, are very hopeful in providing more information.
Dr. Chong, when I'd asked that question, you kind of jumped in. Did you want to add something?
No, I think Dr. O is correct. I mean, when we do the neutralization studies, we're just looking at one aspect of the immunity system, which is probably the one that's easiest to measure, looking at neutralization. But those studies do not look at even the T cell responses or even the memory B cells or plasma cells. So there are a lot of other aspects to the immunity from getting an infection or vaccination that we haven't studied. And those might be just sufficient for us to still have some degree of protection, even though the neutralization studies sometimes sound dismal. It's not to give up hope on the vaccines. They will still convey some degree of protection.
I want to give this opportunity to anybody who's watching on LinkedIn or Facebook. If you have any questions, please put it in the chat and we'll make sure to answer it. And while we're waiting to see if any of those roll in, Dr. O and Dr. Chong, both questions to you. If there's one thing that that you hope that people take away about variants and about mutations, what would it be?
Well, for one, the measures that we're doing, the masking, the social distancing, hand washing, all these good practices, they should still remain right now. I mean, just because vaccination is there, it doesn't really mean that we can drop our guard and be, and start assembling and not taking precautions.
I just want to echo that, and I think the WHO has come out with a statement which really makes a lot of sense to me. The more people who can get vaccinated and the more people we can sort of provide some sort of immunity to, the faster we can stop the virus from spreading and developing more mutations, which would make it harder to treat. It's crucially important at this point to spread the vaccines as much as possible globally so that we're not constantly chasing after variants popping up in various areas of the world, and we can get back to some sort of normalcy. And so vaccinations are crucially important, and social distancing is crucially important at this critical stage.
Right, and to follow up on what Dr. O is saying, exactly, I mean, this effort to eradicate or at least, change the cause of the disease is a global effort. And any vaccine that is out there, whether it's a Sputnik Russian vaccine or the Chinese vaccine or or the Western vaccines, the more people we even vaccinate, the better, because that will build up the immunity, prevent giving the virus a chance to keep multiplying and coming up with new mutations, because it has to be a global effort to control this disease. And especially now with international travel and stuff, this is it's not only in the Western world, we have to make it a global approach.
Thank you both, and we did get some questions that have come in, and they're good questions. So to the vaccine, do you think that this is going to be an annual shot that changes every year because of mutations and variants?
I suspect it would. This is going to be probably very similar to influenza, and we probably have to keep doing this.
And then on a similar note, does it seem likely that people will need to end up getting an annual COVID shot like we do with the flu?
I think likely, yeah. And especially as we noted with international travel, I think one of the biggest things people are missing right now is being able to see relatives and being able to travel. And with the global reach of this disease, really being able to address different variants and try to have as much coverage as possible is going to be to everybody's benefit.
Another question from social media: how long would you advise a person who tested positive for COVID to wait prior to getting the vaccine?
Chris?
Yeah,
There's
I'm
recent
not.
CDC guidelines.
Yeah, there are CDC guidelines out there. So, but I must say I haven't memorized those guidelines.
And they tend to change, so I would check the CDC website.
Right. Well,
information.
we'll make sure the person who asked that question, we'll make sure to get the CDC link with that information on there. And then one last question that's come in, and if you don't have the answer for this, we can follow up on it as well. Is there current testing the vaccine against new mutations in kids under 18?
I don't think we know the answers because the pharmaceuticals are the ones who determine who they are testing it, the population they're testing on. I'm sure they're working on it, but I think since the older individuals were the ones that get the disease worse, that's why they started working on doing trials on that group first.
Thank you very much, Dr. Chong. And those were all the questions that came in. I want to give you guys both the opportunity to say any last thoughts you had about this topic.
Well, I think if you take a look at some of the international websites, the IHME modeling website, they now are including data on what it looks like with the variants coming in and so forth. I just looked at it the other day, and it's interesting because they have modeling of what it looks like if the variants start spreading. They have a line for what if the variants start spreading and our previous infections are not protective. And it also has a line for if there's 100% masking and social distancing, and those curves are markedly different. The curve when people wear universal masks and socially distance looks a lot better. So again, I would urge to not letting up on our vigilance on that front.
Right, I agree, Dr. O. And also, I think we have a lot of heroes to thank for what I've been doing. The people who do the testing, the people who are distributing the vaccines, and the people who are actually now working on the vaccines, because it must be very hard work to keep tracking every mutation and then making all these adjustments. Because I mean, the virus is a very formidable enemy. It keeps changing its tactics, and we got to keep adapting to it.
Dr. Chong and Dr. O, thank you so much for your time today and for joining us and for your insight on this important topic. I learned some things too, so thank you very much. And to everyone who's listening, thank you for watching in, for sending in your questions. To learn more about our initiatives, programs, services, and ways to give, or if you are looking for medical care, you can visit providence.org, and make sure to follow us on social media at Providence Health System for LinkedIn, Facebook, and Instagram, and under Providence on Twitter. Thank you so much, everyone. Have a great day.
Thank you.